So now going over some questions on computing for medicine, right? So firstly, we have the multiple choice questions. I've opened the GPT tab on the right and the questions for this quiz on the left. So we'll go through each question in depth, right? So section A, we have only one correct option. Among the four samples, 1, 2, 3, and 4, which sample or samples exhibit the highest richness and the highest evenness in the distribution of species respectively? And we've been given a table with 4 columns and 4 rows, sample 1 to 4 and species A, B, C, D, right? And we need to find out the sample or samples which exhibit the highest richness and evenness. Okay, so let us first understand how we perform, how we check for this richness and evenness constraint. So which sample has the highest richness and which highest evenness? So richness is basically the count of distinct species or OTUs or ASVs in a sample. A sample with highest richness is the one containing the most different species. So a variety of species, the greater the variety and the greater the richness, right? Now, evenness measures how uniformly individuals are distributed across those species. A sample with highest evenness has species in nearly equal abundance, whereas low evenness means a few species dominate. By definition, a sample with highest evenness has nearly equal abundance of species. Low evenness means a few species dominate. So there is disparity, or few species would be dominating while others are subordinate to those species. Now, by definition, one sample, that is sample 3, can have the most species, right? So if you see, sample 3 has the most number of species, right? When you add these up, no, wait, okay, not add this up, but all of the types, you can see that species A, species B, species C, species D, none of these are 0 for sample 3. But in other cases, there are zeros. That means there are cases where in that particular sample, a particular species out of the 4 does not exist. But in sample 3, we have all types of species. So that means this is the richest. So it can have the most species, that is sample 3. And another is sample 4, can have the most uniform abundance, that is it has evenness, right? What is evenness? It measures how uniformly individuals are distributed across the species. A sample with highest evenness has species in nearly equal abundance, so 15 and 10, right? Now, the sample 3 likely has the most species and sample 4 has the most equal distribution. This matches option C, 3 highest richness and 4 highest evenness, right? So this is the answer. This is wrong actually, right? The explanation given by GPT is wrong because it says evenness. Now, if you see, sample 4 is not at all even, right? Because it does not have uniform abundances of species, 15, 10, and then 0 and 0. So the option given by GPT is wrong. Instead, the answer is D, 3. That is, only sample 3 has the most species, that is the most diversity or the highest richness, right? Most diversity. But 4, that is wrong, right? So D is the answer. Why not the others? Option A, 1 and 3 assume sample 1 has highest richness, but by assumption we infer sample 3 has more species. Only one sample is highest for both metrics. Why are variable regions of marker genes, so now moving on to second question, why are variable regions of marker genes such as the 16S rRNA gene for bacteria commonly targeted for sequencing? So the answer is C, variable regions contain unique genetic sequences that can be used for species identification. So the answer basically lies in the fact that 16S rRNA gene contains hypervariable regions, that is V1 to V9, which are interspersed among highly conserved regions. Primers bind to the conserved flanking regions and amplify the intervening variable regions. These variable regions accumulate mutations that differ between species, providing a unique fingerprint for taxonomic identification. Thus, sequencing variable regions yields species level information. Now, for a specific DNA sequencing, let us complete this. Option C, unique sequences for species identification is correct. Hypervariable regions contain enough sequence diversity to distinguish taxa. Why the others are wrong? Because the answer here, variable regions provide information on total DNA content of the sample. Variable regions do not report total DNA, they report taxonomic composition. Variable regions, the option B is variable regions are highly conserved across all microbial species. By definition, variable regions are not highly conserved. Conserved regions flank them so that primers can bind. And D is basically variable regions are shorter and easier to sequence than other genomic regions. Their length per se isn't the main reason, rather it's their high information content. In short, marker gene studies target hypervariable 16S regions because they maximize taxonomic resolution. Now, for a specific DNA sequencing read, the probability of the base call being incorrect is calculated to be 0.001. What is the Phred score, Q value for that base based on this probability? Now, the Phred value is basically given by Phred quality score, which is defined by Q is equal to minus 10 times log of P error to the base 10. So if error probability is given as 0.001, that is P error is equal to 0.001. What is the Phred quality Q score? So Q score is basically minus 10 log to the base 10 of this value 0.001, that is 10 to the power minus 3. So you bring this power ahead and you get 3 outside, and log 10 to the base 10 is 1. So 3 times 10 is 30. So that is the Phred score. So the answer is 30, Q30. In practical terms, Q30 means 99.9% base call accuracy. So it means 1 error in 1000. Now incorrect options: Q20 corresponds to P0 0.01, Q25 to 0.003, Q15 to P almost around 0.00, approximately 0.03. So Q30 would basically imply this, and that's the answer. Now, which of the following is an example of a reference database that contains ITS sequences? So the answer is UNITE. So the ITS is basically internal transcribed spacer region, which is used mainly for fungal identification. The UNITE database is specifically dedicated to curated fungal ITS sequences. It is the standard reference for fungal ITS-based taxonomy. So these are some types of questions that you have to rote learn. So these are the questions that you cannot derive in the exam unless you know it exactly. Unless you've studied it and memorized it, you won't be able to answer it, right? Moving on, we have the following figure, which represents a rarefaction curve of 16S samples C1 to C10. Based on the curve, which of the following is true? In C1, okay, let us first interpret the graph. So this is the graph that has been given to us: number of OTUs on the y-axis and number of sequences on the x-axis. So interpretation of rarefaction curves: a plateau would indicate that additional sequencing yields few new taxa, meaning entire diversity has already been captured when we reach a plateau, right? So that means it has become constant. So if the graph shows a plateau here, that means the curve is plateaued. That means that additional sequences would yield only very few new taxa. Entire diversity has already been captured, right? But if a curve hasn't plateaued, that is, it is growing, that means more sequencing is required to detect rare species. So the questions options imply various plateau conditions for specific samples. Without the actual curve, one infers if none of the listed is exactly correct, answer D would be applying. But let us see. So it says sample C8 and C9 should be sent for sequencing again to capture the entire diversity, C8 and C9. So that is completely wrong because sample C8 and C9, as you can see, have plateaued, right? So they have reached almost a constant value here, right? They have reached a constant. So this means that their entire diversity has already been captured, right? So what does that imply? That sample C8 and C9 should not be sent for sequencing again to capture entire diversity because it has already been done, right? Now moving on. In C1, C3, and C7, the entire microbial diversity has been captured. C1, C3, and C7, entire microbial diversity has not yet been captured because we haven't reached a plateau in these graphs. So you can see that these are all increasing. So that means we would require diversity. We would require to perform sequencing more to get more diversity, right? So this is also wrong. That is, microbial diversity has not yet been captured completely. Now, sample C9 can be considered as a sample where the entire diversity has been captured. That is true because C9 it has plateaued completely, and that means that the entire diversity has already been captured. So, in general, a sample whose rarefaction curve has plateaued means its diversity is saturated. If the curve still rises, more sequencing is required. Right now, this was a good question. Now, moving on. While analyzing the sequence quality report produced by FastQC for 16S samples, high level of sequence duplication is observed. Why this may not be wrong or erroneous output, and there could be a biological reason for it. So it can be an indicator towards enrichment bias or could be higher abundance of few taxa. So let us understand this problem. Amplicon sequencing, for example, 16S PCR, inherently produces many identical reads from the same region. If a few taxa dominate a sample, the 16S sequences are overrepresented. FastQC uh, reports high duplicate reads, but this often reflects true biological overabundance, not a mistake. Option B, enrichment bias or higher abundance of few taxa is correct. PCR amplification and dominance of common species lead to many duplicate reads. For example, highly expressed transcripts in RNA sequencing or abundant taxa in 16S can yield greater than 70% duplicates, and FastQC warns of duplicates, but for amplicon data that is expected. While others are wrong, bad quality sample and it should be rejected. So duplicate reads do not imply poor quality. Then C option indicates a problem with sequencing instrument and requires immediate technical adjustments. No. Problem with the instrument, no, because duplication here is biological, not an error. And then it is indicative of low sequencing depth and additional sequencing should be performed to increase coverage. So low depth, in fact, low depth usually yields fewer duplicates. High duplication suggests abundant templates. In some, high duplication is a known artifact of PCR bias and abundant taxa. So that is why B is our answer. Now moving on, for sequencing, around approximately 200 BP high quality reads cost upon quality of a fecal microbiome, choose the machine. Let us read the question. I want to sequence the microbiome from a human fecal sample and I need DNA sequence reads that have an average length of around 200 BPs, that is base pairs, but are of extremely high quality. Which sequencing machine should I use for this purpose by considering the cost as well as the quality? So the answer is Illumina. Illumina, why we chose this? Because Illumina platforms, for example, MySeq and NovaSeq sequencing, produce short reads, that is approximately 150 to 300 base pairs with very high accuracy, greater than Q30 for most bases. at relatively low cost per base. They are standard for 16S and short metagenomics. So option B is correct. It yields approximately 200 base pairs reads with greater than 99.9% accuracy, suitable for 16S metagenome sequencing at scale. Now, why not others? Nanopore gives very long reads but lower raw accuracy, around 85 to 95% without extra error correction. Then Pack Bio, that is Pacific Biosciences, can yield very high accuracy long reads, but those reads average thousands of bases, not 200 base pairs, and are costlier. And Illumina is basically optimized for short, high accurate reads, right? So that is our answer. Now I'm running a tool where a division algorithm is working in an iterative manner such that each iteration consists of identifying the unique sequence with the smallest p-value, forming a new partition with that sequence as its center, right? And reshuffling sequences to their most likely partition. Which of the following am I supposed to get through this process? So the answer is amplicon sequence variance, ASV. So this describes the data tool, that is divisive amplicon denoising algorithm or similar method, which iteratively partitions reads by unique sequence, and error model p-value to resolve the exact sequences. The result is a set of amplicon sequence variants, exact sequence variants inferred from the data, right? Now ASVs, not OTUs. ASV basically is amplicon sequence variants, are the output of data to UNoise style denoising algorithms. They are exact biological sequences, 100% identity groups rather than clusters at a fixed similarity threshold. The algorithm uses statistical tests, p-values, and sample error profiles to call true variants. Now, why not others? OTUs are basically operational taxonomic units. They are typically defined by fixed percent identity clustering. For example, 97% by heuristic methods, usearch, vsearch, ucluster, not by iterative p-values. Then species level taxa is also not correct. Output only taxonomy assignment would give the species name for this. Then the iterative divisive algorithm or the iterative division algorithm is exactly how data 2 infers ASVs, amplicon sequence variants. So B is the answer.